Hello guys, welcome to Molecular Biology series. I'm Vineet and today I'll be discussing about the heterochromatin and euchromatin. So if you look about the general definitions, this heterochromatin is highly condensed form of chromatin. This heterochromatin highly condensed and contains lot of gene poor sequences. Okay, so which are not involved in any kind of transcriptions, okay, and also a lot of inactive genes in them, okay. So this is basically the highly condensed version of the chromatin molecule, inside the nucleus. Okay, so heterochromatin is the highly condensed version of chromatin, where the euchromatin contains a lot of active genes. Okay, and also less condensed than the heterochromatin. Okay, if you even look at the typical structure of this heterochromatin and euchromatin. Okay, so these are compact. The heterochromatin is compactly packed. Okay, so this doesn't allow any kind of transcription machineries or the replication machineries to act on the DNA sequences, but this euchromatin allows all the factors or all the regulator proteins to act on the DNA sequences or the gene sequences. Okay, and the major difference between the heterochromatin and euchromatin is the methylation patterns and the acetyl groups. Okay, so the methyl groups, they keep the heterochromatin in inactive state. So that means the DNA sequence to the chromatin to be in the heterochromatin state, okay, or the inactive state, the methyl groups, okay, and the acetyl groups, they keep the chromatin in euchromatin state, okay, or the transcriptionally active state, okay, transcriptionally accessible state, okay. So these acetyl groups, they are meant to keep the chromatin in transcriptionally accessible state, while the methyl groups, this methyl group, okay, methylation patterns, okay, the monomethylations or the dimethylations or trimethylations, they are responsible for maintaining the chromatin in heterochromatin state. Okay, so this is what major difference between the heterochromatin and euchromatin. And beside keeping the genes in inactive states, okay, the heterochromatin also acts as a graveyard for selfish DNA repeats. Okay, so this what the no, this is what the second point says. Okay, so besides acting as a graveyard for selfish mobile DNA repeats, okay, so this heterochromatin also contributes for lot of biological functions such as chromosome segregations. Okay, we have a lot of roles of this euchromatin inside our nucleus. Okay, like maintaining the active DNA sequences, or the maintaining the genes in active states, or making the DNA sequences more accessible for the transcription process or the replication process. Okay, so they are the major roles for the euchromatin. Okay, apart from the euchromatin, heterochromatin also plays a major role inside the nucleus, making the genes to be stay in inactive forms. Okay, so that is what the major role. And apart from that, it also acts as a graveyard for selfish mobile DNA repeats, which you often call as jumping genes, or the 8 rich sequences. Okay, adenine thymine rich sequences. Okay, and multiple features of heterochromatin include the presence or absence of specific histone modifications, DNA methylation patterns. Okay, as I've said you, the methylation patterns on the chromatin. Okay, so these are responsible for keeping the chromatin in inactive state. Okay, that is keeping the chromatin in heterochromatin state. Okay, and small RNAs. Okay, so like the microRNAs or the small nuclear RNAs have been implicated in distinguishing heterochromatin from euchromatin in various organisms. Okay, so these methylations and DNA methylations and small RNAs, they are they distinguish between the heterochromatin and the euchromatin in different organisms. Okay, that is what this third point says. Okay, and cell malfunctions if the genome fails to restrict the repressive chromatin marks with the heterochromatin domains. So what happens inside the nucleus is heterochromatin is spread to one extent. Okay, so that means from one point to some other points, the heterochromatin can be seen inside the cell. Okay, and after this point, okay, after the restriction point, okay, what you call as a restrict point or the chromatin mark. Okay, so after this restriction, you won't be seeing any kind of heterochromatin. Okay, so most of the DNA will be in the euchromatin state. Okay, that is the gene active states. Okay, so this kind of spreading, or this kind of restrictive marks, okay, if they are not present, okay, inside the nucleus, the cell will undergo abnormalities. Okay, cell will show abnormalities, or it malfunctions inside the it malfunctions inside the body. Okay, so this is what some type of this is what the major difference between the heterochromatin and euchromatin. And the spreading is actually maintained by some kind of DNA sequences or the specific elements of DNA sequences, okay, so they are called as insulators, okay. So these insulators, they maintain the spread of this heterochromatin into the euchromatin, okay. Then satellite DNA is often major constant of this centromeric heterochromatin, okay. Centromeric regions of the chromosomes, mostly they contain alpha satellites, okay, alpha satellites and the beta satellite DNAs, okay. So these are what the AT rich sequences, okay. So these are often referred to as the satellite DNAs, okay, and these satellite DNAs, they are mostly present in the centromeric regions, okay, and they are the major constituent for the heterochromatin, okay. If we even look at this image, okay, which is a which is representing the Y chromosomes, okay. Okay, so Y chromosomes in human bodies, it contains lots and lots of heterochromatins, okay. The pink color, wherever the pink color is there, that represents the heterochromatin state, okay. So this one and the centromeric regions of the Y chromosomes, okay, and here these regions actually determines the heterochromatin states, okay, and also the centromere, okay. And we also have ampliconic genes, okay. Ampliconic genes, which increases the turnover number, okay. So of different ampliconic regions, which increase the turnover number of certain DNA repeats, okay. And then we have the X transport regions, okay, which mainly represents some kind of mobile elements, okay, mobile DNA repeats, okay. And then we have the X degenerate regions, okay. And then we also have the palindromic in ampliconic regions, okay. And we have multi-copy genes and the single copy genes, okay. But if we look at this single copy genes and the multi copy genes, they are quite low in number, okay. So we only have low regions of this Y chromosomes containing the single copy and the multi copy genes, while we have a lot of ampliconic genes which increases the turnover number and the heterochromatin states on the Y chromosomes, okay. So most of the heterochromatin material is found in the Y chromosomes, okay. Apart from this, lot of chromosomes also contain the heterochromatins, okay. But mostly this heterochromatin keep the genes in inactive state, okay. That is for the major point about this heterochromatin, okay. And this heterochromatin, it is observed in two stages, two forms, okay. The one is the constitutive heterochromatin and facultative heterochromatin, okay. So the major difference is how much time heterochromatin stays in the inactive form, okay. So that is only the difference between the constitutive and heterochromatin, okay. Whenever you try to observe a cell under light microscopy, the non-dividing cell under the light microscopy, okay, and when you stain this cell, the nucleus of cell when you stain the nucleus, okay, this nucleus appears as the darkly and lightly stained this nucleus appears as under the microscope, it contains some kind of a darkly stained regions and the light stained regions, okay, or dark gray regions or the light gray regions inside under the microscope, okay. So these light gray regions, they actually represent this euchromatin states, okay, and the dark gray regions under the light microscope, they represent the heterochromatin regions, okay. So this heterochromatin is of two types, or stays in two forms. One is constitutive heterochromatin and the facultative heterochromatin, okay. And most of the Y chromosomes is made up of constitutive heterochromatin, which is never gets activated, or which is never get converted into euchromatin state, okay. So this constitutive heterochromatin is the permanent feature of all cells and represents the DNA that contains no genes and so can always be retained in a compact organisms, okay. So this constitutive heterochromatin never gets converted into euchromatin, or never get unfolded into euchromatin or gene active regions, okay. So this is what the permanent one which stays in the cell throughout the life, okay. So this chromatin will be in the heterochromatin state throughout the life, okay. That type of chromatin you call that as a heterochromatin, okay. While the facultative chromatin, it is not a permanent heterochromatin, okay. It can be converted into euchromatin when you remove the methyl groups and when you add the acetyl groups on the specific lysine amino acids on histone proteins, okay. So whenever you do that, the heterochromatin can be converted into euchromatin, okay. So the major difference is what? The time, the period inside the nucleus, okay, where the chromatin, if the chromatin stays in repressed form, or if it may convert into the active form, okay. If it converts into active form, that is euchromatin state, that is called as a facultative chromatin. If it stays in the repressed form or in the heterochromatin state only throughout the life, that at that in that case, we will be calling this chromatin as constitutive heterochromatin, okay. So when these genes are inactive, their DNA regions are compacted into heterochromatin. Okay, it is assumed that organization of heterochromatin is so compact that proteins involved in the genome expression simply cannot access this type of DNA molecule. Okay? Because the heterochromatin itself is a highly condensed region inside the nucleus. Okay, if it is highly condensed, lot of proteins won't have enough access to gain the enough access to go and bind to the DNA sequences. Okay, or the major groups of the DNA sequences. Okay, if it is loosely bound, the DNA is readily available, the promoter regions of the DNA gene sequences are readily available, so the proteins can interact with this promoter sequences. Okay, if the chromatin is highly condensed, just like this constitutive heterochromatin, these promoters cannot access the DNA sequences easily, or the promoter sequences easily, and cannot bind to these promoter sequences. Okay, making that the genes to stay in the inactive form only. Okay, so that is what the difference between the hetero and euchromatin. Okay, so how can we maintain this the heterochromatin states or euchromatin states inside the nucleus? Okay, there are, as I said you, there are certain type of process. Okay, methylations and acetylations. Okay, these kind of methylations and acetylations makes a not certain type of regions inside the nucleus or the chromatin regions, okay, to be in the heterochromatin or the euchromatin states. Okay, so the methylation of the both or DNA or specific sites on histones, it is a feature of inactive chromatin. Okay, so that means whenever you perform these methylations, okay, methylation reactions, okay, by using certain type of enzymes called as SUV39H1, which is a Z domain containing enzyme, okay, Z domain containing protein, okay, or enzyme. So this enzyme actually adds a methyl group on 9th position amino acid, that is the lysine amino acid, on histone 3. Okay, so histone 3 contains lysine amino acid on the 9th position. Okay, so if it contains a methyl groups, then it is an indicator of heterochromatin. Okay, in order to perform the methylation, first you have to remove the acetyl group which is present on the lysine amino acid. Okay, so this lysine acetyl group on the lysine 9 amino acid, okay, 9th position lysine amino acid, is an indicator of euchromatin state. Okay, and if you are removing this acetylation, that means the deacetylation process is happening. Okay, and whenever you try to add a methyl group on the same position, okay, so that marks the heterochromatin state. Okay, so this is what this is what the major enzymes to keep is responsible to keep the chromatin in heterochromatin state. Okay, and then we also have a lot of other proteins called as heterochromatin protein 1, okay, HP1, which is also a histone chaperone, or the histone couriers. Okay, courier proteins. Okay, so this is HP1, which is also a histone chaperone. Okay, so it also binds to the 9th amino acid, that is a lysine amino acid on histone 3, with its chromo domain. Okay, so it also has a domain. Okay, and by binding to this histone protein, okay, it adds the methyl group on the histone protein on lysine amino acid. Okay, and it also convert the euchromatin into heterochromatin. Okay, so this is what the major enzymes which are involved in maintaining the heterochromatin states. Okay, and then HV1 then targets the activity of DNA methyl transferases. Most of the methyl sites on the DNAs are CpG islands. Okay, so we know about the cytosine per guanine sequences, which we often call as the CpG islands. Okay, so they contain lot of methyl groups on them. Okay, so cytosine contains a methyl group on them. Okay, so this CpG islands also plays a major role in epigenetics. Okay, so these CpG islands, they are also they are often present in these heterochromatin regions. Okay, so these CpG islands, whatever the methyl groups are there on cytosine nucleotides. Okay, so they are actually transferred by the DNA methylases. Okay, DNA methyl transferases, they transfer the methyl groups onto this sequence, onto this cytosine sequences which are present in the CpG islands, and makes the chromatin to be in the heterochromatin states. Okay, and I said you about the insulators. Okay, so insulators, they stop the spreading of this heterochromatin into the euchromatin. Okay, so these are specific DNA sequences, or some kind of elements, okay, inside the DNA. So they act as a barriers for the spread of this heterochromatin into the euchromatin. Okay, so they will be presenting in the middle of this heterochromatin and euchromatin, and they stop the spreading things. Okay, and these insulators, they are known to contain some kind of a special sites known as the hypersensitive sites. Okay, so these hypersensitive sites are specific to these insulators. Okay, and these hypersensitive sites, okay, and mostly they are present in the promoter rich sequences, okay, wherever the gene contains, wherever the promoters are present in for a particular genes, okay. So these promoters, often known to contain this kind of hypersensitive sites, okay, apart from the insulators, okay. So these hypersensitive sites, they are short regions of chromatin. That means, the DNA sites only, the DNA sequences only, short regions of the chromatin that are sensitive to cleavage by nucleases like DNase 1, okay. DNase 1 is a nucleus that cleaves the DNA, okay, and they are known as DNase 1 hypersensitive sites, often found near or within promoters, okay. So why they are present within promoters or near the promoters? Because they can get easily accessible to the a lot of other transcription machineries, so the DNA replication machineries, okay. The factors, if they want to bind to this sequences, DNA sequences, some kind of accessibility should be there for these factors, okay. That accessibility is actually provided by this DNase 1 hypersensitive sites, okay. And these hypersensitive sites are majorly or specific for for these insulators, okay. Insulators are also DNA sequences, okay, and which are also regarded as the special chromatin structures, okay. So they are tend to contain these hypersensitive sites, okay. They mark the spread of this heterochromatin into euchromatin, okay. They prevent the spreading of heterochromatin into euchromatin, okay. So this is what about the insulators, and this is how you keep the heterochromatin in heterochromatin state only, okay. So we require only methyl transferases, okay, methyl transferases proteins, okay, which contains the chromodomain, okay, and just like this HP1 protein, and also a lot of DNA methyl transferases, okay, and also the set domain containing enzyme, which is also called as a SUV39H1, okay. So this type of proteins or enzymes, they make the chromatin to be in the heterochromatin state only, okay. So this is about the heterochromatin. Then we have this euchromatin. So euchromatin, I've said you already, this is a less condensed DNA, less condensed regions of the chromatin, where a lot of gene active regions are present, okay, and lot of DNA sequences are easily accessible to the factors which are involved in the transcription process or the gene regulation processes, okay. So the exact organizations of this euchromatin inside the nucleus is not known, but some parts of the gray regions, light gray regions, okay, lightly stained gray regions are known to be in euchromatin states, okay. These regions are believed to be in euchromatins, okay. And these euchromatin specific chromatin sequences, they are present in the nucleus in the form of loops, okay. So these kind of loops, they are about 40 kb to 100 kb in length, okay, and they are predominantly present in this present in the 30 in the form of 30 nanometer fiber and associated with the nuclear matrix, okay. So these sequences, the euchromatin sequences, which are forming the loop structure about 40 to 100 kb base pair length, okay. So they contain a lot of adenine and thymine rich DNA segments, okay. These adenine and thymine rich DNA segments, okay, so they will now go and bind to the nuclear matrix, okay. So this kind of binding is also regarded as this kind of binding is also tells you that there is some kind of euchromatin is present inside the nucleus, okay. So these loops which are attached to the nuclear matrix via this AT rich DNA segments called as a matrix associated regions, okay. So these matrix associated regions which are present inside the nucleus, okay, these are also called as a scaffold attachment regions, SARs, okay, which I have discussed in the genome organizations, okay. So they make the chromatin in euchromatin states, okay. So this euchromatin actually seen in the case of loops, okay. So this is actually present in the form of loops, okay, loops of DNA. So these loops of DNA are about 40 kb to 100 kb in length, okay, and these loops are known to contain adenine and thymine rich sequences, and these adenine and thymine rich sequences, they are bound to this nuclear matrix called as matrix associated regions, okay. These regions, these sequences are known to bind and maintain the euchromatin state, okay. And we also have a lot of other proteins, okay, so which maintain the deacetylation states, or which perform the which transfer the acetyl groups onto the histone proteins or the lysine amino acids, okay, to maintain the euchromatin, okay. If these acetyl groups are moved by the removed by the deacetylation process, the euchromatin can again get converted into heterochromatin, okay. And recently the Jumanji proteins, okay, so which are discovered in Saccharomyces pombe and Saccharomyces pombe, Arabidopsis thaliana, and filamentous fungi Neurospora casa. Okay, so these Jumanji proteins, they bromodomain containing proteins. Okay, so they are discovered in these organisms. Okay, so they are responsible for protecting the euchromatin from heterochromatin mox. Okay, what they do is they transfer a acetyl group to the lysine amino acids, or they also remove the methyl groups from the chromatin to make the chromatin easily accessible for the factors which are involved in the transcription process. Okay, that is maintaining the euchromatin state. Okay, so these Jumanji proteins or JMGC proteins, okay, they are subfamily of this 2-oxaloglutariate oxygenases. Okay, and these proteins non-heme iron or 2-oxoglutarate dependent oxygenases. Okay, and they have a characteristic antipallar beta sheet structure. Okay, so this beta sheet, you can call that as a super secondary structure or a certain type of modems. Okay, or specific modems. Okay, so these proteins, these contains this antipallar beta sheet structure. By using this, they bind to the DNA sequences and remove the methyl groups on the DNAs or the histone proteins. Okay, so they are known to demethylate the histones. Okay, methylated histones will now undergo demethylations. Okay, and this kind of demethylations makes the heterochromatin to get converted into euchromatins. Okay, and we also have another member of this demethylase family. So this Jumanji proteins also belongs to the demethylase family only. So the second member is also known as the founding member. This demethylase protein is also family protein is also called as lysine specific demethylase. Okay, so it is a flavin dependent amino oxidase that targets mono or dimethylated lysines. Okay, not the trimethylated lysines. Okay, so this removes the methyl groups on the histone 3, specifically this fourth lysine amino acid on histone 3 protein, or the ninth lysine amino acid on the histone 3 protein. Okay, so this specifically removes the methyl groups on these lysine amino acids. Okay, so these are two major proteins which are involved in making the heterochromatin into euchromatin, or which are mainly involved in maintaining the euchromatin states inside the nucleus. Okay, so this is what the major roles of this demethylases. Okay, we have demethylases and deacetylases. Okay, deacetylases are responsible for keeping the heterochromatin in heterochromatin state only, and demethylases, they are responsible for maintaining the euchromatin in euchromatin state. Okay, so this is what about the two different types of proteins. Okay, and there are also a lot of other remodelers which you also call as the chromosome remodels or the chromatin remodels. Okay, so these chromatin remodels, they are also known to keep the euchromatin state only. Okay, so this euchromatin, only some euchromatin is transcriptionally active, but not all the euchromatin is transcriptionally active. Okay, if you want to make the euchromatin transcriptionally active, okay, you have to do it in 3 ways. Okay, so what are those 3 ways? So first one is what? Positioning of the nucleosomes. Okay, so you can slide the nucleosome from one region, one specific region to another specific regions by using these chromatin remodelers, or you can also you can also do it by replacing the histone with their variants. Okay, the histones also contains a lot of variants. Okay, so you're replacing the actual histone molecules with the histone variants. Okay, actually the histone 3 contains a lot of variants. Okay, whenever you try to replace one of the variant with this normal histone 3 proteins, you can convert this heterochromatin into euchromatin. Okay, and we also have this covalent modification just like this demethylations. Okay, demethylations or the acetylation process. Okay, whenever you add an acetyl group for the specific lysine amino acid which is present in the histone protein. Okay, covalent modification, so you can make this heterochromatin into euchromatin. Okay, so this is what the chromosome remodeling is. Okay, so all this process, they are collectively called as a chromosome remodeling, and there are a lot of other proteins are there inside the nucleus which perform this chromatin remodeling. Okay, so 5 known families of enzyme complexes actively reposition or displace nucleosomes, hydrolyzing the ATP in the process. So 3 of these are particularly important for the transcription activations. Okay, so I have gave you 5 different family members. Okay, 5 different families of this chromatin remodelers. Okay, one is the histone modification complex. Okay, and we have switch and SNP proteins over here, and one is a ISWI family. Then we have SWRA family, and then we have HERA. HERA is also known as a molecular histone chaperone. Okay, this SW and SNF, switch and SNP. Okay, switch and SNP. Okay, these proteins, they are mainly responsible for the nucleosome remodeling. Okay, that is the sliding moment. Okay, so they slide the nucleosomes. Okay, from one region to another regions like this. Okay, this is called as a nucleosome remodeling. So I will be discussing all this process in the gene regulation topic. Okay, and then we have this ISW1 family. This ISW1 family is also involved in this nucleosome remodeling and also transcriptional repressions. Okay, switch and SNP is involved in the transcriptional activation, while this ISWI family is involved in the transcriptional repressions. Okay, it's exactly opposite of this switch and SNP protein functions. Okay, and this FWR family. Okay, so which contains 12 monomeric subunits in them. Okay, 12 polypeptide chains in them. Okay, so this is an oligomeric protein. Okay, which is found in the eukaryotes. Okay, so this is involved in the variant deposition, histone variant depositions. Okay, H2AZ variant of H2 histone. Okay, so this is involved in the depositing of this histone variant. Okay, I said you about this histone variant, right? When you deposit an histone variant in the place of actual histone, that also makes the euchromatin state. Okay, that also marks the euchromatin state. Okay, and then we also have histone chaperones, HERA. Okay, which also involved in the deposition of this histone 3 variant, that is histone 3.3 during the transcription process. Okay, whenever the transcription process is happening, so this HERA actually replaces the histone 3 with its variant histone 3.3 to make the DNA sequences readily available to the factors involved in the transcription process. Okay, so these are about some families. Okay, histone remodelers, so chromatin remodel families. Okay, and we also have GCN5, ADA2 and ADA3 family. So this is about a tripeptide sequences. Okay, so these are majorly present in the yeast. Okay, and lot of other chromatin remodelers are mainly discovered in the eukaryotes only. So this yeast, okay, contains GCN5 as histone acetyl transferase activity. Okay, so these proteins, they transfer the acetyl groups to the histone molecules. Okay, if you transfer the acetyl groups also, if we transfer the acetyl groups, that also converts the heterochromatin into euchromatin. Okay, that means gene accessible states. Okay, so this is what this is what the major things about the heterochromatin and euchromatin. Okay, just remember, heterochromatin is mainly maintained by the methylations. Okay, so a lot of methyl transferases, they transfer the methyl groups, okay, onto the histone proteins. Okay, specifically lysine amino acids. This marks the state of heterochromatin state. Okay, while euchromatin, okay, it will be seen inside the nucleus. Okay, and it is marked by having this acetyl groups. Okay, so CH3CH2 acetyl groups. Okay, so these acetyl groups, if they are present on the lysine amino acids, if they are covalently bonded to this lysine amino acids on the histone proteins, okay, they mark the euchromatin states. Okay, and we also have a lot of chromatin remodelers. Okay, like switch and SNP proteins, or GCN5 proteins, or HERA proteins. Okay, which might change the entire histone protein, or which might add methyl acetyl groups on the histone proteins. Okay, or which might displace the nucleosome to different regions. Okay, so that causes the conversion of heterochromatin into euchromatin. Okay, so this is what the basics of this heterochromatin and euchromatin, and in the next class we will be discussing about the histone variants and their epigenetics.